Good evening, and on behalf of SSR, I'm very pleased to welcome you to today's webinar, Novel Contraceptives Targeting Sperm Development and Function. My name is Janice Evans from Purdue University, and I'm a member of the Contraceptive Symposium Organizing Committee, and I will moderate today's webinar, and today's session co-chair is Dr. Amelia McKenzie of FHI 360. And in kicking this session off, the first of this three-session conference, I very much would like to acknowledge all those who made this possible. So the Symposium Organizing Committee was chaired by Daryl Russell, and the other members have been John Hennebold, Diane Duffy, and Ozgi Fazlabas, with Andrea Cupp as past president of SSR, also helping us out along the way. Ozgi Fazlabas gets some special recognition as he was a key point of contact for us when I was SSR president in the year that we were putting together an ask to the Bill and Melinda Gates Foundation. And indeed, they've been wonderful in providing the support for some programming at the 2019 SSR annual meeting and for this special virtual conference with the goal of advancing contraceptive research. SSR also has been very pleased to broaden our reach with the help of an international partner, and that's the International Congress of Endocrinology. So we're very excited to welcome participants. I think we have over 300 registrants, which is fantastic, from all areas of reproductive biology research in all geographic regions to engage in this exploration of challenges and opportunities for new innovations in this really important and underrepresented field of study. And then finally, let me just hit on a few logistics before we begin. So all participants have been placed on mute, and you'll be on that throughout the call. If you have questions, we'll ask you to submit your questions using the questions and answer feature at the bottom of your screen. Please do not use the chat feature for the Q&A. We very much want you to use the question and answer feature. Also, you can upvote a question that you see if you like that, and that'll elevate it in our prioritization for the Q&A session. Questions will be read at the end of each presentation by Q&A facilitator and co-chair Dr. McKenzie. So additionally, the webinar is being recorded and will be made available to all members on the SSR website. So with that, I'm very pleased to kick things off with our keynote speaker. We excitedly welcome Dr. Martin Matzuk from Baylor College of Medicine, who will be presenting his work in this important area with his talk entitled DNA-Encoded Chemistry, Technology, and Functional Genomics for Contraception Research. So welcome, Marty. So pleased to have you with us.
Okay, do you hear me okay?
Hear you great, thank you.
Okay, all right, well, thank you very much for the nice introduction, Janice. It's really a pleasure to give this lecture, and I also thank Daryl Russell. I don't know if he's online, but if he is online, it's 5:30 a.m. in Adelaide. I also want to thank SSR as well as the Bill and Melinda Gates Foundation and Stephen Ward for the support of this symposium. So this symposium was supposed to happen initially in Argentina. Instead, I am located in the middle of the Texas Medical Center. We're at Baylor College of Medicine. My building is right here in the middle of the medical center. I'm not sure if you can see that thing. I was asked to start your video.
Are you mentioning your pointer? Can see that just fine.
Oh, okay, that's fine. I got a message saying that the host is asking me to share my video, so I just want to make sure that everything is okay. So anyway, we're located in the Texas Medical Center, and this is the world's largest medical center. There are two approaches that we're taking to validate contraceptive targets. The first is a genetic approach, and the second is a chemical approach. In the case of the first approach, we're using CRISPR-Cas9 to mechanistically discern key reproductive tract proteins and there are pathways required for these various processes of spermatogenesis, sperm function, and or fertilization. The second approach that many of you are probably not as familiar with is called DNA-encoded chemistry technology. In this case, we're utilizing a strategy in which every single molecule in our library is covalently linked to a piece of DNA, and the DNA has a barcode which tells us what that molecule is, and I'll go through that in a few minutes. So we're using DECTech to find these initial small molecule probes, say preclinical candidates, to target druggable proteins essential for these processes that we've identified in aim-1. And then the third aim is then to evaluate in vitro efficacy and in vivo contraceptive effects of these drug-like inhibitors. So our population is now over 7.8 billion. And as you can see here from 1960 until the present, this is essentially a straight line. However, as you can imagine, this is putting tremendous stress on our planet, and it's believed that the Earth is reaching a tipping point. And global population growth has been called a wicked problem. And so Dr. Sunny Ramaswamy in 2014 gave a speech in which he called a booming world population the mother of all wicked problems. And a wicked problem is one that is difficult or impossible to solve because of incomplete, contradictory, and changing requirements that are often difficult to recognize. So, and he also went on to say that the clock is ticking, and it's important for us to address this challenge and do it in the next few years. So the wicked problem lacks a current fix, and there's no single solution to this problem. So again, we must act very quickly. You're seeing that species extinctions on land and in water are happening at a very rapid rate. It's believed that we're losing large amounts of mammals, birds. In the seas, we're losing coral reefs, which are necessary for the survival of fish. And the window of opportunity is small, but we still believe that we can do something to save species and populations. And the problem caused by humans must be solved by humans. So because there's no single solution to this wicked population growth problem, we believe that we are pursuing multiple different points of interrupting conception. And we're using this DECTech, or DNA-encoded chemistry technology platform, to help investigators find hits that can possibly become new chemical entities in the future. And we've initiated a number of collaborations related to male contraception. This includes Debbie's group at UNC, Irv at Northwestern, and Joy at Washington State. We also offer our contraceptive research community an entry point to address the wicked population growth problem with new therapeutic options. And we've been lucky enough to get support from this and to be encouraged for these kinds of collaborations through NICHD, the Bill and Melinda Gates Foundation, the Walsh Foundation, and CPRIT, which are both in the state of Texas and are supporting our library synthesis, as well as the Male Contraceptive Initiative. So to validate our targets in mice, we have this go, no-go strategy. And one of the first steps in this process is to determine whether or not the mice are fertile. So either you have a fertility defect or the mice are fertile. If the mice are fertile, the next step is to halt the analysis and publish the results. So over the last 5 years, we've worked together with Masahito Ikawa's group at Osaka University, Thomas Garcia's group at Baylor College of Medicine, and my group also at Baylor College of Medicine. And we've first identified over 120 genes that are dispensable for male fertility. And our first publication came out in 2016. And then we subsequently added on to this list of genes that are not required. And this year, we've also published 2 papers in our special issue of Biology Reproduction that just came out last month, in which we added another 23 genes to this list. And so you can go online, and all these papers are available online with open access. So, so the next step is, if you have a fertility defect, what we like to do is then define the defect. And then the next step after that is then, once you define where is the defect in the process, you want to define the molecular mechanism of action and the signaling pathway. And so to do this, we've utilized, again, CRISPR-Cas9 to insert tags into alleles. And this allows us to look at the protein localization. If necessary, we also generate antibodies to the proteins. Many of the proteins, because of course they are focused within the male reproductive tract, there's not usually typically antibodies to these proteins. We will also go ahead and identify protein interactome using these tag proteins. So we could pull down with the tags, because again, if the antibodies aren't available, we want to utilize pull downs. And if we insert bio-ID into the loci, we also want to use the bio-identification system to be able to also find local proteins that are localized very close using proteomics analysis. And lastly, perform additional in vitro and in vivo functional studies on these knockouts, and eventually complete the analysis and publish the findings of either a single knockout or multiple knockouts that are in related pathways. And I'll give you a few examples of them today. So one of the first processes that I like to talk about is the sperm acrosome. And so the acrosome biogenesis is a really critical process for male fertility. The acrosome is on the head of the sperm, so here would be the tail of the sperm. Here would be the acrosome, and the acrosome. You can see the acrosome here on the head. The acrosome reaction then releases the acrosomal enzymes and antigens, which then go ahead and wind up resulting in allowing the head of the nucleus to be visible. So, so in the process of doing these, just a little bit about acrosome biogenesis. So in contrast to secreted proteins or proteins that wind up on the membrane, the proteins go from the ER to the Golgi to the surface of the nucleus to form these acrosomes. And a number of proteins which are important in vesicular trafficking, wind up being important. We wind up also seeing that some of the proteins, such as one of the additional proteins, ZPBP1, was knocked out, and we were able to find that the knockout of ZPBP1 results in this round-headed sperm, or globozoospermia. In addition to SPACA1 knockouts, this was a group this was, sorry, I'm getting texts here in my office. So SPACA1 was identified by Dr. Fujihara in Masahiro Ikawa's group in Japan, and this was published in 2012. So in addition to these proteins which we identified previously, what we were able to find is additional proteins such as SSMEM1 or SPAR1. And SSMEM1, which I'll talk about in the next slide, is a very important protein in which this protein appears to play an important role in the localization and the movement of the Golgi apparatus, and we're still continuing to work on that. So, so this was work from Dr. Kaori Nozawa. She is a postdoc in my group. She previously had done her PhD in Masahiro Ikawa's group. And this is a collaboration on the knockout. SSMEM1 is the name of a protein called serine-rich single-pass membrane protein 1. This was published also in a special issue of Biology Reproduction last month. So when we knocked out SSMEM1 using CRISPR-Cas9, what we're finding is that in contrast to the normal, this is a scanning EM picture, in contrast to the normal heterozygous mice, what we find, which is a sickle cell shape, we're finding that the knockouts have these globozoospermia or round-headed shape. If we turn to transmission electron microscopy, we can find that the heterozygotes during spermiogenesis wind up having this nice formation of the acrosome very tightly linked to the nucleus. But in the case of the SSMEM1 knockouts, we wind up seeing that we still continue to see the Golgi localized in the wrong place, and so there's mislocalization of the Golgi. And if we map this out, normally we can go ahead and the Golgi winds up at the certain stages to move very quickly towards the bottom of towards the tail of the sperm. In the case of the SSMEM1, this Golgi winds up remaining, and we're continuing to figure out exactly why that process is occurring. So in addition, once we find that we have mice that are sterile, if these proteins we believe are druggable targets, then they can then become contraceptive targets. So the next step in the process is to generate recombinant proteins and then perform a drug screen with DNA-encoded chemistry technology. So when I started the Center for Drug Discovery, one of the important things was to create libraries with very unique compounds. And so the high throughput screening collections that you often find in industry, often those libraries, even though there's large numbers of molecules in those collections, first of all, they're very expensive to get together. And second, many of the molecules may look very similar, even though you have many of them. So for example, even though these apples are different colors, they're all very similar apples. However, what we wanted to do is we wanted to create as diverse a collection of molecules as possible. So typically in industry, you wind up having high throughput screening collections or about 1 million compounds. We wanted to generate many more molecules and with much more diversity. And you can see here, similar to these fruit, we want to have molecules of different sizes, shapes, charges, etc. So when we decided 5 years ago to start working on DNA-encoded chemistry technology, or DECTech, we decided to really come into this, and I'll tell you exactly why. So in this case, similar to the name implies, all of the molecules are attached to DNA. So here we have the first building block here, which would be this green hexagon. We would covalently link this green hexagon onto a polyethylene glycol linker, which is attached to DNA. Then on the other side of the DNA, we would then ligate on a piece of DNA, which would be an 11-mer piece of DNA, which would totally correspond to this building block. And at any single position, for example, in his case, we can have up to 1,400 different building blocks, and we could have 1,400 different 11-mer sequences, which would correspond exactly to each building block. Then we can add a scaffold. In this case, we can then add our second building block, shown here as this purple building block. We can ligate on the second 11-mer sequence. We can then covalently link a third building block to the structure, a third codon, this 11-mer sequence, which would then correspond to this red. So this green, purple, red sequence then would be acting as a barcode, which would tell us exactly what this molecule is. In addition, on this end, we can then add additional DNA, which is very, very important for marking each individual library. So what that does is it allows us to be able to mix all the molecules together when we're doing the screen, because each molecule, of course, has a barcode associated with it, just like when you go to a supermarket and you go through the cash register. Each grocery item has a barcode that tells you what that item is. In addition, because each library has additional sequences that tell us what it is, we can then mix all the libraries together when we're doing it. And this large number of diverse compounds then allows us to screen and enhances the probability of finding these highly potent hits. And our DECTech library synthesis team includes Yingqiu Chen, Dewey Du, Barry Lee, Surinder Dewadi, and Nihan Uchicek. So to date, we've now created 52 different libraries containing a total of 5.6 billion unique compounds. And the libraries can be screened as a pool in most desired combinations. We design these libraries with structure-activity relationships in mind. They have many different types of molecules in any individual library. Also, the CDD has a team of geneticists that we've assembled, biologists, cheminformaticians, chemists, and crystallographers that help us with the validation, DECTech screens, and further work up with these small molecule hits and leads. So these are, in the process of doing this, we've developed a lot of our own on DNA chemistry. Because when we're generating these libraries, because there's DNA present, we have to make sure that we do not destroy the DNA. we have to also make sure that we don't destroy the other molecules that are attached to DNA. And so we've, as I mentioned, developed a number of technologies in-house at the Center for Drug Discovery at Baylor College of Medicine. We've also optimized a number of additional reactions that were developed by others to make them easier for us to work with. So this is just the pathway for how we actually do this tech. So as I mentioned, the ability of us to build these various libraries and mix them together for our selections. So all the selections are done in a single individual tube or tubes. And the next step is to have a tag protein. So all of the selections are done in a single test tube. We then go ahead and do the screen in which the protein is tagged. Typically, we like to have a histidine tag on the protein. The protein is then subjected to selection of all these libraries together in a single tube. We then are looking for, in the library, we're looking for this protein to find the molecules that interact perfectly with it. We then go ahead and pull out these molecules with the attached DNA with magnetic beads, so nickel magnetic beads. And we go through 3 rounds of this selection to enrich for this molecule with the DNA attached. We then, next step is to sequence the DNA. By sequence the DNA, we can go ahead and do the decoding. And with the decoding, we can then find out what the molecules are that matches that barcode. And of course, we get in any individual sequencing run, we get many different kinds of molecules, many of which are related, as you'll see in a few seconds. Once we do this, we can then go ahead and resynthesize the hits off the DNA and then do the additional studies: hit conformation, structure-activity relationships, metabolism, pharmacokinetics, toxicology studies, etc. In addition, we've developed crystallography strategies in the group, and we also utilize our NMR and drug metabolism core, which is associated with our Center for Drug Discovery. So this is just a little bit of background for one of our targets, BRDT. The molecule that we utilize in this case is JQ1. JQ1 work was a collaboration with Jay Bradner's laboratory when he was at Dana-Farber Cancer Institute. So we published this work 8 years ago, and what we're able to show is that JQ1, the plus and the enantiomer of JQ1 has very nice activity, whereas the -1 does not. The small molecule binds nicely in this pocket when crystallography was done. And we actually subjected mice to treatment with JQ1. We find that it reduces testis weights, sperm counts are reduced, and motility of the sperm are reduced. So it has both a decrease in sperm counts as well as a decrease in the function of the sperm. So using this information, we want to go ahead and test our libraries to make sure that we can actually do these studies. And so Buffone, you and Nick Simmons did these experiments within our group. Buffone runs our selection team. He's assistant professor here at Baylor. And Nick was a former postdoc in our group who's now moved on to industry. So here's a histidine tagged BRDT protein. Nick was able to go ahead and link the JQ1 molecule to DNA. And then we were able to test whether or not we could find this piece of DNA during the selection process. So if we do 2 selections, this is a qPCR analysis. If we do selections, 2 rounds of selection in which we do not add our BRDT protein, we don't add our histidine tag BRDT protein, we do not get any enrichment. But if we add our his tag BRDT protein and do the enrichment, after 1 round of selection, we get about 100-fold enrichment. After 2 rounds, we get another 100-fold enrichment. And if we would guess after 3 rounds, we would get a 1,000,000-fold enrichment total. So that's telling us that allows us to get rid of the molecules within our libraries that are not binding to BRDT, but do select for the molecules that are binding very selectively to BRDT. So this will be a typical experiment in which we would have, in this case, if we had an inhibitor present for any different protein, we would have a 3-tube assay. Tube 1 would contain all the libraries that we have. We would add no protein and no inhibitor, and so that would just tell us whether or not we had any molecules binding to our beads. The second tube would have our BRDT protein present, and it would lack an inhibitor. So that would be telling us, do we have any molecules that are binding specifically to BRDT? In the third tube, we would have BRDT protein, but we would add an excess of our competitive inhibitor, JQ1. And so if these molecules that are binding in tube 2 are binding to the same site as JQ1, JQ1 should block that binding. So this is the informatics analysis that was done by John Faber in our group. He's a cheminformatics faculty member in our group. And if he maps the enrichment for BRDT, so this would be tube 2 versus tube 3, you see that we get this significant enrichment for a number of molecules. And all these molecules are binding to the same site as JQ1, because if we add JQ1, we don't see any of them being sequenced. So this is what we've done. And now if we take and we do a real, if we then take their analysis and we're looking for whether or not we can find selective compounds that bind to either the first bromodomain of BRDT or the second bromodomain of BRDT, we find that in fact we can. And so if we're looking first at this alpha screen assay, this BRDT alpha screen assay, so this is the first bromodomain, this is our alpha screen assay for the second bromodomain. And what we see here is we have this red line is this CDD956 compound. So it has a very nice 2 nanomolar inhibition, IC50 of 2 nanomolar in this assay. But in the case of the BRDT, the second bromodomain, we see very poor binding. We also had molecules like CDD991, which were very good at binding to a second bromodomain. This is a 6 nanomolar inhibitor. But in fact, it bound very poorly to the first bromodomain. And then this magenta compound shown here is a compound that bound equally to both the first and second, and it binds very similar to JQ1, which is also shown here in black. And this is work that was done in the group by John Jin, a former staff scientist, Andrew Ku, a staff scientist, and Justin Englund, also staff scientist in our group. So once we go through the selection, the hit triage, and the X-ray crystallography is one of the next steps in the process. And in parallel, we're typically analyzing the various studies, the various molecules, the lead compounds using ADME and toxicity. So this was a collaboration with Chol Kim, who's a member of our center. He's a crystallographer, and Rajesh Sharma in his group. And so these are 2 of the potent CDD molecules that were binding to BRDT, second bromodomain, and we were able to get very potent compounds at 2.3 angstrom and 1.9 angstrom resolution as co-crystals. In addition, one of the next steps in the process was to move on to additional proteins, and one of the questions that we had was, could we find inhibitors of isozymes? So for example, LDHA is a somatic protein, and whereas LDHC is a testis protein. So these various LDH isozymes of LDH1 through 5 and LDHC, which is the testis-specific protein, wind up being in various places. And these proteins have fairly similar homology. They have about 75% amino acid identity. But LDHC, because it's in the testis and because of the work that Irv Goldberg has done for most of his career, has shown that this is very relevant to contraception. And the reason why we thought that LDHC could be targeted using this strategy is because a compound from GSK was found to bind very potently to LDHA, but we were able to show that it doesn't bind very well to LDHC. So using this DECTEC approach, we're able to then screen our LDHC protein for enrichment for LDHC versus enrichment for LDHA. And we found some molecules, such as the CDD1277 molecule, which was bound well to LDHC, but didn't bind well to LDHA. CDD1279 bound a little bit better to LDHA, but still was very potent at binding to LDHC. And when we analyzed this data, these molecules off of DNA, what we're able to find is that the CDD1279 molecule that I show right here, it is a 1 micromolar inhibitor, whereas binding to LDHC is a 0.3 micromolar inhibitor. Whereas the 1277 molecule bound less potently to with lower affinity to LDHA, still had a very high affinity for LDHC. And so we had a 25-fold enrichment for binding this molecule to LDHC over LDHA. In addition, the next area that I wanted to touch on was protease-focused libraries. And so this is work from Surinder Dewadi in our group. And what Surinder was trying to do was generate libraries that were very similar to known protease inhibitors, such as this argatroban. So he wound up generating these libraries, these 3 cycle libraries, and the steps are all shown here. The idea of these focused libraries is to really build operational capacity to support this emergent action. So if we decide that there's a protease out there that we want to hit, we can go ahead and try to do that. And so this paper was just published a few months ago, and we used thrombin as our control. And in the process of making these libraries, this is in the paper, we generated a 9.8 million compound protease bias library in a paper, and we also followed it up with a 258 million molecule library. And so we used thrombin as our proof of principle that this library strategy was working. Argatroban in this assay is a 9 nanomolar inhibitor, and this kinase assay, or sorry, these protease assays were done by Kurt Warren in our group. And CDD 1472 turns out that we pulled out of libraries is a 1 nanomolar inhibitor. So using this strategy, we could find even more potent inhibitors than the current best thrombin inhibitor out there. This also allows us then to utilize these libraries for various purposes, such as there's many proteases that are important for in infectious diseases. We also have a number of various healthcare conditions, such as male contraception, that we want to go after. In the process, there are male and female reproductive tract specific proteases that we're interested in, and we identified those through our CRISPR-Cas9 approach. So a little bit about the migration through the uterotubule junction. So this is the UTJ. So this is, so as the sperm are released into the female reproductive tract, they migrate into the uterus, and then eventually they'll have to get through the uterotubule junction, enter the oviductal isthmus, and then go ahead and fertilize the cumulus oocyte complexes within the ampulla. And in the special issue of the special BOR issue, we were able to, we had 2 papers published, both for proteases. PRSS55 is a protease, TMPRSS12 is also a protease, and again, in collaboration with Massa's group. We also have another paper that we just published in Science a few months ago, in which we targeted another protease, OVCH2. And then working with Yoshi Fujihara, Massa's group and my group, we were able to identify a number of other male reproductive tract specific proteins that are important for this process. So again, we have three proteases that wind up being targets that we want to go ahead and screen. OVCH2 is very unique in that it's expressed only in the epididymis, and I'll show you that data in a few seconds. TMPRSS12 is a transmembrane protease, only testis-specific, and PRSS55 is a GPI-anchored protease on the surface of the sperm. And LYPD4 in the PNAS paper that I just pointed out, Yoshi was able to find this protein is also a GPI-anchored protein, but not a protease. So just to show a little bit more, so these proteins that we identified go along with a large collection of proteins that have already been identified by Masuro Okabe and Masahiro Ikawa at Osaka University. And so PRSS55 is newly identified along with TMPRSS12 and OVCH2, as I mentioned in the epididymis. So if you look at OVCH2, it's made as this secreted, epididymis secreted protease, and it has a signal peptide consistent with most secreted proteins. It has these cub domains, which we believe are very important for localization of the protein within the epididymis. And the antibodies that were generated in Osaka by Kiyosumi, who's the first author on the paper, have shown that OVCH2 is localized very specifically in this region in the head of the sperm, and is critical for maturation of the sperm. So if you look at atom 3, which is important marker for processing of the sperm, atom 3 is not processed well in the OVCH2 knockout. In contrast to controls where you see lots of these marked sperm, these red marked sperms, in which you see the sperm entering the oviduct, you wind up seeing, in contrast to the knockout, fails to see the sperm entering the oviduct. So just to finish up, so we have also a strategy in which Ying-Chu Chen and Barry Lee have developed other libraries that are kinase biased to doing similar strategies for kinases within the reproductive tract. And just to summarize, our DECTech team and our platform, we've assembled this team of geneticists and DECTech members for doing these various studies. Novel chemistry and scaffolds have been used for generating 52 libraries of over 5 billion compounds. This allows us to identify these drug-like molecules to related human and non-human proteins. I'd like to also just bring up the fact that I mentioned a few times during the presentation, the special issue of Biology of Reproduction. Gunda Georg, Wei Yan, and I are editors of this special issue called Contraceptive Development, Past, Present, and Future. With the help of the Bill and Melinda Gates Foundation, the Male Contraceptive Initiative, and SSR, this whole issue has now become fully open access as of today. You can go to this special issue. There are 30 papers, including our editorial, reviews, commentary, forums, and research articles. And they cover various aspects of drug development and include a number of active investigators and experts in drug discovery. So finally, I'd like to acknowledge individuals at Baylor that are PIs: Thomas Garcia, Chol Kim, Damian Young, Diana Monsivice, also Feng Li over here. I'd also like to mention that we have key external collaborators, Masahiro Ikawa, who, when this picture was taken, his center here, and was visiting our laboratory. We also have Yoshi Fujihara, who did a sabbatical in our group and is now at the National Cerebral and Cardiovascular Center in Osaka. Debbie, Irv, and Joy I mentioned during the presentation, and then more recently, Sung-Hee Baek at Seoul National University. And I have to thank the Eunice Kennedy Shriver and ICHD for all of their help and support since I first got my grant, my first grant in 1991. The Bill and Melinda Gates Foundation, which has been critical for our studies of male contraception. Male Contraceptive Initiative, which has recently helped us in a number of key ways, as well as the Welsh Foundation and CPRIT. And I'd like to finish and thank you all for your attention by being online today.
Thank you so much. What a great summary of your really interesting work that your group and collaborators have been doing to really move the work in contraceptive R&D field forward. We do have a couple of questions here that I'll start by asking. And the first is, do any of the acrosomal phenotypes in mice phenocopy known to idiopathic human male infertility cases? And actually, I'll just kind of add on as a little bit of a broader question, like how do you integrate human infertility phenotypes into your go and no-go decision algorithm?
Of course, thank you very much for the question. And let me see if I can actually share my screen here. Maybe I can't share my screen. I think I'm having problems sharing my screen. Anyway, let me see if I can do this. Anyway, thank you very much for the question. So, we are starting to identify more and more of these various phenotypes which have an acrosomal biogenesis or globospermia phenotype. And those are really critical now for understanding whether or not they play roles in patients with that. We haven't done enough sequencing of the patients, and I'm hoping that as we produce a lot of these various knockouts, that more studies will be done in patients with these phenotypes.
Great, thank you. The next question was, are there any plans for searchable database for the dispensable proteins? And I'll just add a little bit of my own ad lib as the prerogative of the moderator. How do you see the libraries being used by the wider contraceptive R&D community?
So we do everything in-house because, as you could imagine, with all the funds that we've invested and all the various people that are important in this process, it's not easy to just give out libraries. And there's lots of studies. It's also very expensive to be able to do a lot of these sequencing. And the easiest is to have experts actually doing all the selections, all making the compounds off of DNA. If you typically give someone a library who's a biologist, they might not be able to have the resources to do the next generation sequencing and have everything else that's necessary. So we set everything up as a collaboration, an equal partnership for doing these studies.
Great, thank you. The next question is, what criteria do you use to define a target as a druggable target?
Yeah, so I think a lot of times if you think of enzymes, I think of those as the classic druggable targets. And so if you look at drugs that are in patients, there's large number of those small molecules may be targeting G protein couple receptors, enzymes, etc. So when I think about druggable targets, I usually am thinking about those various classes. So I mentioned a few of those cases, proteases and kinases, as being some of those various druggable targets. Proteins that I think are poor targets for any kind of selection are proteins that are somewhat malfolded, just normally. They have large amounts of regions that are not typically in a nice conformation.
All right. I have two questions about the BRDT work. Have you identified viable alternatives to JQ1 for use with contraceptives? And then can you tell us a bit more about BRDT as a contraceptive target, since it targets spermatogenesis early on, could be effective, but perhaps likely to be reversible?
Yeah, so let me just go after the second question first. So with regard to BRDT, we think that it is, when we did these studies in mice, the mice were reversible. So we can give the drug, we can result in infertility, we can stop the drug, and we can go ahead and the mice revert. So the fertility was reversible. So we think that in men, it would also be the same thing. The problem has been, for everyone working in the field, is that there is a somatic protein, a BRD4, and that's how JQ1 was initially identified. And so there appears to be a lot of the drugs that have entered the clinic for targeting BRD4. They've found on-target effects of BRD4, which are limiting their usefulness, whether for cancer or non-cancer. So that even though we've used as our proof of principle, we think that, even though we have very potent small molecules, some of which are more selective for BRDT, we have to be worried about on-target BRD4 on-target effects of any kind of male contraceptive. That's true of any single target that we're working on. We have to be worried about in normal men or normal women, you and especially young individuals, you have to make sure that these young individuals are, whatever you give to them, that they don't have side effects, because of course that will be very dangerous for them.
I think we have time quickly for one last question here that's been upvoted. Have follow-up mating and fertility trials been performed as part of the drug development pipeline for the most promising candidates?
Yes, we've begun to do some of those studies, and we've had some nice results for those studies.
Great. Well, with that, I'll hand it back over to Janice to introduce the next talk.
Thank you, Amelia.
Thank you both very much, including for the interesting questions. Hopefully some of them that could also be part of an end of session discussion. So we'll continue now with 3 additional talks selected by the organizing committee. The first of these will be Laura Giojalas from the National University of Cordoba in Argentina. She'll continue on this theme of thinking about sperm physiology, but here with a focus on sperm chemotaxis. And her talk is entitled Sperm Chemotaxis and its Potential Application for New Contraceptive Development. So welcome very much, Laura. We look forward to your talk.
Thank you. First of all, I very much appreciate the invitation to participate in this meeting, especially to you, Janice. Okay, first, I would like to define what is sperm chemotaxis. When a molecule is distributed as a gradient, a spermatozoa can sense this gradient and can orient their movement according to the direction where it is the source of this molecule. For instance, if the attractant source is located here, and a spermatozoa can sense this gradient of the attractant, we call chemoattraction, that most of the people know it as chemotaxis. So I will talk about it, chemotaxis, instead of chemoattraction mostly in my talk. But when the source is a repellent molecule, a spermatozoa sense this gradient and go away from the source of the attractant, and this is called chemorepulsion. So what do we know about the sperm chemoattraction? It was observed in several mammalian species, including human, in a small sperm subpopulation around 10 to 20%. They have to be capacitated and acrosome intact. Several attractant sources and molecules has been identified, and I will refer especially in this talk about progesterone a little bit. The chemotactic movement is expressed firstly by a linear movement, then to search the source of the attractant, the sperm make a turn, and then the pattern of movement become transitional. The molecular mechanisms of chemotaxis involve the EAC pathway, the GC pathway, calcium mobilization, and protein phosphorylation. Sperm chemotaxis is transient, so it lasts the chemotaxis behavior about 2 hours, and it shows periodicity. I will talk about some of these results in this short talk. We were interested in progesterone because it was secreted by the cumulus cells by the time of ovulation. And this steroid can attract spermatozoa. So to test that, we ran a very large dose response of progesterone concentration, and we found out that at picomolar range, there was the highest level of chemotactic spermatozoa, which was similar to the positive control follicular fluid. We further depleted the oocyte cumulus complex condition medium from progesterone with antibodies here, or by immunoprecipitation, and in both cases we observe a significant decrease in the level of chemotactic spermatozoa. Concerning the sperm chemotaxis periodicity, I like these results because help to design our experiment. For instance, we incubated under capacitated condition spermatozoa for 24 hours, and every hour we determine the percentage of chemotactic spermatozoa, and we observe the cyclicity with valleys and peak along the time, and most of them last around 2 hours. And this is called ultradian rhythm. But when we determine chemotaxis every month along the year through 3 consecutive years, we find out 2 peaks per year, one in autumn and the other in the spring, where the level of chemotactic spermatozoa were maximum. And this kind of cyclicity is called infradian rhythm. So this sperm chemotactic cyclicity is important just to design our experiments and avoid negative results. Concerning the molecular mechanism, independent of the attractant, for instance, oral substance like Burchonal or progesterone or nitric oxide or atrial natriuretic peptide, most of them are sharing the same signaling to conduct to chemotaxis. For instance, starting with the AC activation pathway with protein phosphorylation in tyrosine, calcium mobilization, the activation of GC pathway, which led to the protein phosphorylation in serine threonine, a second intake of calcium, and those can lead to sperm chemotaxis. We recently find out that progesterone activates the NOX5 that is located in the plasma membrane, and these generate ROS, and these ROS in turn activate the AC pathway. Considering that capacitated spermatozoa can be selected by a 10 picomolar gradient of progesterone, we design a sperm selection assay that consists in 2 wells connected in the base by a tube. In one of them, we place spermatozoa previously capacitated under capacitated conditions, incubated under capacitated conditions, and in the other one, we place 10 picomolar progesterone. Therefore, a gradient of progesterone is forming from well 2 to well 1. When we collect the sperm from well 2 after the assay, the sperm population is enriched in capacitated sperm, intact with intact DNA, low oxidative stress, they are viable and motile, the number of bound sperm to zona pellucida is increased, and embryo cleavage and quality is improved. We use it just for several mammalian species, fresh and frozen thaw samples, sex and unsex samples, and we are currently carrying on a clinical trial in humans. What do we know now about sperm chemorepulsion? We observe in several mammalian species, also in a subpopulation, a small subpopulation. They have to be capacitated. Several repellent sources were identified, like those that are artificial, like progesterone receptor ligand, as emergency contraceptive, like ulipristal acetate, like UPA, mifepristone, and levonorgestrel, or natural lysine that is released immediately after fertilization. The chemorepulsive movement and the molecular mechanisms are very similar to that described for chemotaxis. For a control, we use progesterone as a positive control, and we have a sperm accumulation in well 2 after the assay, but when we put in well 2 UPA instead of progesterone, we got a negative accumulation of spermatozoa in well dot. Look at the very low concentration that is elicit, also in the picomolar range. And what means that spermatozoa are skipping from well 2 to well 1. In a more complicated assay, when our control also, we got a sperm accumulation in well 2, but when we put homogeneous distribution of UPA in this chamber, together with the progesterone gradient from well 2 to well 1, we also observe sperm chemorepulsion. This means that the chemotactic progesterone gradient was converted in a repellent one. We then tested SIN for repulsion, since, as I said previously, it is released upon fertilization, and we found out the control the same, and we put zinc together with progesterone. We found out also repulsion to zinc. We further incubate with or without UPA, and then perform IVF, and we found out a significant decrease in the spermatozoa that arrive to the egg. So sperm chemotraction and chemorepulsion can be considered as the two sides of the same coin, because they share the small sperm subpopulation, they are capacitated, they respond to gradients of low attractant or repellent concentration, and they share the similar molecular signal and pattern of movement. Here we have a model where in the center it is drawn an oocyte surrounded by the cumulus cells. On the left side we have an unfertilized oocyte where cumulus cells are secreting progesterone, forming a concentration gradient which can be sensed by capacitated spermatozoa to find out the egg and fertilize it. After fertilization, immediately after, zinc is released from the egg, where in an environment where progesterone gradient it is also present. So under this situation, capacitated spermatozoa can be repelled from the egg. But in this case, where a non-fertilized egg also is surrounded by the cumulus cells and secreting the progesterone gradient, under the presence of one of these PRA, capacitated spermatozoa can be repelled from reaching the egg. Therefore, by manipulating this sperm behavior, fertilization may be prevented for contraceptive purposes. Many thanks, that's all. I think it's a little bit more than 10 minutes.
Thank you very much. We do have time though for a couple of questions, and they're related actually to the rhythms that you described in the beginning. The first is, why do you think that you see the infradian rhythm, the different responses during the year, and how many hours of incubation and capacitation media was used for that graph that shows the variation during the year? And the second question related to that was, how conserved are those rhythms across species?
For the first one, you refer to the seasonal one. We incubate the spermatozoa for at least 4 hours every month. In the middle of the month, we perform this assay with several human sperm samples. And why 4 hours? Because of the logistic to work with this sperm sample, we use 4 hours because we know that at 4 hours we have an acceptable level of sperm capacitation. And the second one question?
How conserved across species are those rhythms?
I didn't get it. How conserved
across species are the rhythms? Like, do different species exhibit those differently?
Different species, you mean? No, we perform it in only with human samples. Okay. And for instance, for reproductive technology, it is important to see if you are performing this treatment for infertility in a time of a year that you will get very low levels of capacitated cells, what is mean Chemotaxis cells, because they are correlated. There is the same state that they need to be capacitated to chemotactically respond.
Thank you. I think we have time for one more quick one, and that is the sperm that have been prevented from fertilize. The sperms that have been prevented from fertilizing an egg, can they fertilize other eggs?
We did it with only one egg per IVF. So we did not use several eggs for the same IVF.
Okay, maybe time for one more. Did you see any impairment in the overall sperm motility in the presence of UPA or zinc?
No, because this is a routine control that we use to test motility, because if the spermatozoa do not move, we cannot evaluate nor chemotaxis nor repulsion, because we need they move to evaluate this, just to see how they orient themselves.
Thank you. I'll turn it back over to Janice for the next talk.
Welcome.
Thank you very much, Laura. We'll continue on this sperm theme, moving back to the acrosome, which was also noted by Dr. Matzuk. Our next speaker is Mariano Buffone of the Institute of Biology and Experimental Medicine in Argentina. And the title of his talk is Sperm Aquasome Reaction and also Capacitation and their potential application for new contraceptive development. Welcome, Mariano. Pleased to have you with us.
Okay, can you see the screen? Yes, right?
Yes, we can. Thank you.
Right. Well, thanks SSR and Janice and Daryl for this invitation. And so today I'm going to talk about like a very special process that is very important for fertilization. We all know that the mammalian sperm needs to be capacitated for fertilization. And this involves complex signaling cascades, both in the head and in the tail, with these signal cascades, sperm can undergo changes in motility, like hyperactivation, and also undergo a very important process for fertilization called acrosomal exocytosis. That is, it's a special type of controlled secretion that is, and because it only occurs in sperm, it's an interesting target for contraception. It's a single secretory vesicle that multiple fusion points between the outer acrosomal membrane and the plasma membrane form these hybrid membrane vesicles. And we don't have like membrane recycle, so this process is completely irreversible. And it's critical for fertilization for two main reasons. One is because if sperm do not undergo acrosome reaction, they cannot penetrate the zona pellucida, but also because many of the proteins that are important for sperm egg fusion cannot relocate to the site for where the sperm fuses with the egg, like ISUMO1, for example. And so during capacitation, many things changed and prepared the sperm to undergo exocytosis. And among all these changes, I'm going to focus on membrane hyperpolarization, and that occurs during capacitation. And the membrane potential is established when you have different concentrations of electrogenic ions across the plasma membrane. And in the case of the sperm, the most important one is potassium. That is, the concentration is higher inside the cell. And if you use the Nernst equation to calculate or to estimate the membrane potential, you can see that during capacitation, the inside of the cells becomes more negative, from 45 millivolts approximately to 65, 70 millivolts. And the main responsible for this change in membrane potential is the slow 3 channel discovered by Celia Santi some years ago in mouse. But in humans, the story is a little bit more complicated, because it is postulated that possibly other potassium channels are also present on the CFTR and ENaC system. So membrane potential in humans is very important. And this is a study that we did in collaboration with Dario Kraft this year. And we basically divided the patients with good IVF rates and poor IVF rates. And we can see that in those couples where the IVF rates were like good, like higher than 60%, most of the patients display hyperpolarization during capacitation. And in those who fail to have a good success in IVF, none of them show hyperpolarization. So it's very important for fertilization. And how is this connected with the acrosome reaction? Well, we can in non-capacitated cells, we can manipulate the membrane potential using different drugs. And you can see here that these are non-capacitated cells that after incubation with any of these growth drugs, amiloride, genistein, or valinomycin, you can induce membrane potential hyperpolarization. And when non-capacitated cells are exposed to, for example, agonist of the acrosome reaction, in this case is the solubilized zona plusia, they don't undergo acrosome reaction. However, if we treat these cells with any of these compounds, that now the cells are ready to undergo acrosome reaction. So this means that the hyperpolarization is necessary and also sufficient to prepare mouse sperm for acrosome reaction. So our question is, we asked two questions in this work. Is hyperpolarization necessary and sufficient to prepare human sperm for acrosome reaction? And what is the mechanisms by which this change in membrane potential prepares sperm to undergo acrosome reaction? And so we use two conditions. One is time zero, which is sperm after swim up or isolate gradient. These cells are non-capacitated, of course, and 6 hours, the cells were incubated under non-capacitating and capacitating condition. This is omitting bicarbonate in the capacitation, in the incubation medium. And you can see the differences. This is a fluorescent method to estimate the membrane potential. And you can see like this in the non-capacitated cells, right after swim up, you have 51 millivolts. And after capacitation, the cell becomes more negative. So then we use, in most of our experiments, we use a potassium ionophore, valinomycin, that produce like the efflux of potassium from the inside to the outside of the cell. And this results, of course, in hyperpolarization. So in these conditions, cells are non-capacitated. As you can see, this is protein tyrosine phosphorylation. After 6 hours, this is one of the hallmarks of capacitation. If we don't add bicarbonate, we don't have protein tyrosine phosphorylation also after swim up. And valinomycin in these conditions did not affect our tyrosine phosphorylation levels. So we tested many different agonists like calcium ionophores, potassium, and progesterone in all the experiments, but because we don't have like too much time to explain all the conditions, and I want you to focus here at time zero, this is right after the swim up separation, where we separate all the motile sperm. As you can see, these cells are non-capacitated, so if we add progesterone, we don't have a good number of sperm that undergo acrosomal exocytosis. However, if these cells were pre-incubated with valinomycins to induce hyperpolarization, now the cells can respond and undergo acrosome reaction. And because valinomycin also alters the membrane potential of other membranes inside the sperm, we also use a different compound, amiloride, that is specifically blocks the INAC channel and also results in hyperpolarization. And basically, we obtain similar results, very poor response in non-capacitated cells, but when these cells were treated with amiloride, now they are ready to undergo acrosome reaction. And so our conclusion is that hyperpolarization is sufficient to prepare sperm for acrosome reaction as it does in mouse sperm. Now, the next experiment, we incubated the cells with high potassium. In this situation, the efflux of potassium that occurs during capacitation cannot occur, and as a consequence, hyperpolarization is blocked. And what we observe here with progesterone or ionophore is that capacitated cells in the normal potassium concentration, they can undergo hyperpolarization. They respond to the agonist of the acrosome reaction. However, in the presence of high potassium, the cells cannot undergo. In these two conditions, we block hyperpolarization. So now we wanted to know how membrane potential hyperpolarization prepares sperm for acrosome reaction. And our candidate here was to look at calcium. And we did that at a single cell level. And we used two probes. One is fluo4 to measure intracellular calcium levels. And the other one is the probe FM464 that increase the fluorescence. This dye increase its fluorescence when sperm undergo acrosome reaction. Here you have one example. This is a cell that increased intracellular calcium and then now increase the FM464 fluorescence because the cell is undergoing acrosome reaction. So what we noticed at the beginning is that cells, human sperm under these are non-capacitated cells, display different intracellular calcium patterns in non-capacitated, when they are non-capacitated. For example, here you can see one cell that is, the calcium is going up, then down, up and down again. This is like a typical oscillatory pattern, but we observe like different patterns of calcium that we classify them in three. One is the oscillatory, or where you can see these clear calcium oscillations, a transitory where the calcium goes up and then goes down, and then the sustained, that is calcium goes up and then, stays high. So what we notice is that when in non-capacitated cells, most of the cells are oscillating their calcium levels. But when we add valinomycin to produce the hyperpolarization, and you can see here, that most of the cells are undergoing like these calcium oscillations. However, when we perform the when we add valinomycin to produce this hyperpolarization, the cells stopped oscillating and now they switch to the sustained pattern of calcium. They increase and then they stay like in higher levels compared to the basal. So hyperpolarizations change the dynamic of intracellular calciums in, of course, non-capacitated sperm by switching from an oscillatory to a sustained pattern of calcium. And what happens when we add progesterone? Well, when we add progesterone—
Mariano, I'll just chime in. Your time's running a little tight, so are you wrapping up?
Yes, one more
Thanks.
slide.
Yeah, gracias.
So when we have these calcium oscillations in the non-capacitated cells, we add progesterone and we have a single peak, but the cells still are oscillating. However, when we add valinomycin, now we have a single peak or a sustained peak, and the cells now they do not oscillate because of the valinomycin. So we wanted to know what's happening when we observe both things together, and we use also FM464. As I show you in the previous slide, we observe in our controls, we obtain similar levels of acrosome reaction at the single cell level. And what we found basically is that those cells where we add progesterone and they are, you have single peak and then you have these oscillations, do not undergo acrosome reaction. When we add valinomycin and now this the oscillatory pattern like goes away, we have this transitory pattern that undergoes acrosome reaction similar to the sustained. But in all the cases, none of the cells with the oscillatory pattern undergo acrosome reaction. Most of them undergo acrosome reaction with the transitory pattern of calcium. So my take-home message is membrane potential hyperpolarization is necessary and sufficient to prepare a human sperm for acrosome reactions. Human sperm, the hyperpolarization suppresses the calcium oscillations, and this oscillatory pattern of calcium prevents the occurrence of acrosome reaction in non-capacitated cells. So thanks for all the group and these great collaborators, in particular Alberto Darson and Dario Kraft and Paulo Visconti. And thanks for the agencies for funding.
Great, thank you. I have a couple questions for you. The first is, how do you see the channels here specifically being targeted for contraception, and could that be done with potentially no side effects in the female, possibly male as well?
Yeah, it's a very good question. I mean, ideally we need to find a high throughput method to find like drugs or molecules that can block one of these channels. The problem is that some of these channels are not sperm specific, so it's hard to not to have side effects in this if you want to block them.
Great, and could you remind us of which specific channels you're thinking are mediating the hyperpolarization?
Well, the for sure, slow 3, the potassium channel, still is under some controversy. Slow 1 is also participating in the membrane potential on human sperm, and also the system, the CFTR ENaC channels that work together.
Great, thank you. I think we are at the end of our time here, so I'll hand it back over to Janice.
Thank you.
Thank you, Mariano. So for the third of these shorter talks selected by, invited by the organizing committee, we'll now have a talk that'll focus on spermiogenesis and some aspects of cytoskeleton biology, with a focus on the homeodomain interacting protein kinase 4. So we welcome Aaron Crapster from Stanford University to talk about these selective inhibitors for male contraception targeting HIP4. Welcome, and we look forward to your talk.
Thank you, Janice. Hello, everyone. Should be able to see my slides now?
Sure can, thank you.
Great. Yeah, so my talk today is going to focus on the development of small molecule inhibitors for a protein kinase called HIPK4, and these would be useful for male contraception. And the first half of the talk will be just reviewing some work that I did as a postdoc at Stanford University, where we discovered and validated HIPK4 from the biology through genetic models of HIPK4 knockout in the mouse. And then the second half, I'll switch to the work that we've been doing at the company that I now work for, which is called Vibliome Therapeutics here in Bozeman, Montana. So homeodomain interacting protein kinase 4 is, it's a dual specificity kinase. It's part of the HIPK family, which includes 4 different kinases shown in the kinome tree here. Let me get my laser pointer on. And, but it's a bit of an atypical member of the family. It actually lacks the homeo, didn't like that. Technical errors. Okay, great. Can you guys see my screen again?
Looks like you're back on track.
Okay, thank you. So HIPK4, it's an atypical member of this family in the fact that it actually lacks the namesake homeo interacting domain portion of it. So it's a cytosolic protein kinase, dual specificity, and it's similar to the DYRK superfamily of kinases in general. But its biological function was largely unknown until recent years when we published in eLife this past summer. So I got interested in the
Aaron, real quick, we have a request just for you to speak a little louder if possible.
Sure. Let me try to boost my microphone here. I guess technical issues are plaguing me. So I'll try to speak a little louder. It looks like PowerPoint is breaking down. Apologies for this. Okay, I should be back online, and I'll breeze through the biology a little bit quicker than I would have otherwise. The main point what we found, this was again back during my postdoc days, was HIPK4 is predominantly expressed in the testes. There's a very small level of expression in the cerebellum, but the predominant expression, as you can see in these in situ hybridization in both mice and in man, is in spermatids. And so looking at different staged seminiferous tubules in both juvenile pups as well as in adults, we were able to limit both the mRNA expression as well as the protein expression for HIPK4 kinase within the round spermatid stage, and maybe some very early elongating spermatids at what would be stage 8 or step 8 spermatids. And using targeted gene knockout approaches, we were able to develop or generate a colony of knockout animals. And you can see here that in many fertility pairs, the male knockout HIPK4 animals mated to either wild type or heterozygous females were completely sterile, while the female knockouts were totally normal. The data aren't shown for the females, but hets can have normal fertility while the knockouts were completely infertile. And if you look at the sperm from these animals, those HIPK4 null sperm are also incompetent for IVF. So shown here is just, these data were produced by the Stanford Transgenic Tumor Model Center, and we obtained no fertilized embryos from IVF procedures using knockout sperm, but using ICSI in a couple different rounds, we were able to generate fertilized embryos at a rate that was consistent with their historical records. And then these could be implanted to generate live pups in a recipient female. So the problem with the sperm and the phenotype that I characterize HIPK4 knockout animals by is oligoasthenoteratosospermia, OAT, would be the classical human condition for that. So I'm showing you here nothing about the motility or the concentration in the epididymis, but you can see through scanning electron microscopy that the head shape is extremely defective in not only the surface topology, but also just the morphology, rather than the sickle or crescent shape of a normal sperm head shown in the wild type case, but we have a range of defective phenotypes here seen with the knockout sperm from knockout animals. And using some fluorescent staining, you can tell that the acrosome is still present in a couple different examples of sperm with similar phenotypes to the electron microscopy images. And I'll also mention, but not show the data, that these sperm can capacitate, and they also can undergo the acrosome reaction, but just at lower rates than wild type. There's also an increased tunnel staining with sperm in the epididymis from knockout animals, indicating most probably an increased rate of apoptosis for these cells, maybe explaining the lower concentration. And if you take a step back further, what's really going wrong is these critical morphology changes at steps during spermiogenesis, which is a cytoskeleton regulated process where the round spermatid change into an elongated form. And so I'm just highlighting with the TEM images shown here a couple of the key features in sperm, the acrosome, and how it should be coupled very tightly to the manchette at the perinuclear ring and the groove belt. And you can see these defects start to, well, some spermatids had completely disrupted step 8 spermatid, but the ones that did make it through showed us these cytoskeletal defects right at the groove belt, where there's a separation from the acrosome and the manchette, which ultimately leads to dysregulation of the sperm head shape as it matures. So that's the background, sort of validating the target and also explaining a little bit about where the biological process is occurring that we would try to be disrupting. And I think a late stage spermiogenesis target is a good one for contraception, and we can get into that later in any question series. So now I'll just switch to, for the sake of time, focusing just to explain how the company here at Vibliome is approaching developing small molecule inhibitors to really kinases in general. So we're a private drug discovery company, and we're a structure-based design team. So we're designing compounds that are exclusively focused right now at inhibiting kinases in a specific way. And if you look at the classical examples of how people describe kinase inhibitors in the literature, there's 4 binding modes or types. And so this would be type 1 through type 4. And the type 1 inhibitor is a classic kinase inhibitor where you're inhibiting or binding with the kinase active site at the same location that ATP would normally bind. So these cartoons are illustrating how the molecules would be binding in these different types of inhibition modes. And there's an activation loop that in many kinases gets phosphorylated to activate it, that would be in what's considered a DFG in motif for an active kinase and a DFG out motif for an inactive kinase. And the main difference between a type 1 inhibitor and a type 2 inhibitor is type 2 inhibitors bind in the DFG out motif, which is an inactive kinase conformation. Type 3 and type 4 are largely considered allosteric kinase inhibitors. And the difference is just how far away from the active site these compounds are binding distinguishes between type 3 or type 4. So type 1 and type 2 interact with the ATP binding pocket, but also type 2 binds in a motif where the enzyme is inactive and it reaches over into a different part of the protein as well. So if you look at modeling of the difference between the DFG loop and this ATP binding site and the type 2 binding site that opens up, which we call the selectivity site, this is how the types of structures that we're comparing when we model small molecule ligands for kinases to distinguish between the interactions that they're making and how they're binding. And so again, the ATP binding site type 1 inhibitors, those are a little bit more challenging to get exquisitely selective inhibitors out of because the binding site of most kinases in the ATP pocket is largely conserved. And so there's not a lot of selectivity that you can gain from tweaking the molecules here if you're hitting more than one kinase, for instance. And of course, in the contraception field, you really want to knock down the function of a single gene product is the goal for safety reasons. So if you look in the literature at known HIPK4 ligands that are inhibitors, pacritinib is one of these, and using these models, you can quite readily see that as shown here in the image with the DFG in loop shown in mauve, that this is binding in a type 1 conformation. And we at Vibliome have some structure-based guiding principles to help us get type 2 binding compounds. And as I mentioned, that can come with some advantages, especially when it comes to selectivity. So our workflow for how we're developing inhibitors, I think, is important to point out from the drug discovery pharma aspect of this, because we're interested in not just the biology. We're interested in developing this further for actual in vivo use. So obviously the first steps that we undergo for all of our compounds is profiling them against the kinome, which at this point we start with around 400 kinases, and then we can profile as well against mutant panels. But then those are followed up with real IC50 determinations at several different ATP concentrations to really get a hint at the potency and potentially the binding mode of our compounds. We're also then interested very much in whether or not our compounds are active in cells as we move towards further towards in vivo studies. So we take advantage of Brett-based technology where you overexpress a HIPK4 fused to a luciferase construct and outcompete a ligand inside of a real cell for that. We also undergo many preliminary and early ADME tox type of studies. These are cell permeability assays, commonly known as the Keiko-2 bidirectional assay. We look at the stability of our compounds in plasma, protein binding in plasma for 3 different species, as well as some off-target potential liabilities in safety screens. Now, I think when you're targeting a protein in the testis, one study that we have underway just now that we're just getting going is looking at whether or not your compounds can cross the blood testis barrier. This is one of the strictest junctions, biological barriers in the body, and it's an important one to try to make sure that if your compound needs to function in the seminiferous tubules, you're actually assaying for molecules that can get beyond that. And so to do that, we would sample fluid from the radi testis efferent ducts over time after injection of our compounds. So like I said, these studies are undergoing, and you combine that with early pharmacokinetic studies to look at what dose you might need to have an effect. So one of the last slides I have is I just wanted to highlight how truly selective our compounds are. And this is all based on modeling, computational docking, and structure-based insights. And at this point, just using homology models of HIPK4. And then SAR and medicinal chemistry efforts to really learn about what features in our molecules are interacting with, potentially with what features in the binding pocket in a type 2 mode. And so you can see here, these are the results looking at 3 different concentrations of the 4 inhibitors that I've shown, and the percent inhibition of 400 kinases. So we've got these compounds that are just really, really selective for inhibiting HIPK4 as well. And these data would translate and do translate to IC50s in the nanomolar range, 1 to 10 nanomolar. And these compounds are then tested, as I mentioned, inside of cell-based assays. And we're seeing these translate to IC50s or target engagement in cells between 20 and 200 nanomolar. So we're really having compounds that are quite selective and that have cellular permeability and activity. And we're testing them now after those early battery of ADME-T tests in vivo settings for fertility studies. And that's where our program's headed right now. Sorry for the technical difficulties, but happy to answer any questions about how we're thinking about the fertility studies that we're planning or anything else, really. I do want to acknowledge the folks at Stanford that helped me during the biology work, and then Gary Flynn and Koi Hung here at Vibliome, who are the brains behind the medchem and compound design.
Great, thank you. I think we have time for a quick question here. Posed by an audience member. I wonder whether male patients treated with kinase inhibitors for different reasons, such as chronic myeloid leukemia, etc., show a similar type of alteration that you found with the HIPK4 knockout mice.
So can you repeat that one more time?
If patients treated with kinase inhibitors for other reasons, like chronic myeloid leukemia.
Yeah, that's a good question. Right. So in some cases, yes, that is true. Although I don't think that many of the oncology trials are looking at that as an endpoint in elderly or even in chronically ill patients like that. Fertility is usually not assessed, but in early studies of before those trials, reproductive toxicology is always an important step. And for instance, if you look at a super promiscuous kinase inhibitor like sorafenib, it's unknown whether or not that compound crosses the blood-testis barrier, but there are several examples in animals where they have a defect in male reproductive.
Great, and one more quick one. Can you explain a bit more about how you confirm that the compounds are crossing the blood-testis barrier?
Right, sure. So if a compound crosses the blood-testis barrier, that means that it's crossing from the outside of the seminiferous tubules to the adluminal space or the lumen inside the seminiferous tubules. That fluid is constantly then pumped out of the radiotestis through the epididymis to eventually the vas deferens. And so what we do is we microcannulate the efferent ducts of the radiotestis, and then with a very small needle. This is work done at down at a small company that spun out of UT Southwestern called Genome Design Laboratories. So in rats, which have larger efferent ducts than mice, inject the compound intravenously, and then over time sample the radiotestis fluid as it's getting pumped out of the testis. And that tells you, and then look at the concentration of your compound in the radiotestis fluid compared to plasma levels of your compound in circulation. And that tells you how much is crossing the barrier.
Great, thank you. There are some more questions, but I think we do have to move on to the next presentation, so I'll hand it off to Janice.
Thanks.
Thank you, Erin, and thank you, Amelia. We have 3 additional short talks. These were selected by the organizing committee from our abstract submissions. And so I'll make a quick note that we have a reminder about joining our virtual happy hour at the end of this 3-session symposium, so 3 Wednesdays from now on October 14th, and that's where we're announcing the winners of the oral and poster presentations. So to kick off the first of these 3 short talks from the abstract submissions, I'll introduce Alan Adam Silva from Sao Paulo State University to talk with us about epididymal protease inhibitor EPPIN. Take it away, please.
Thank you, Janice. I'm going to share my screen just a moment. Can you listen and see my screen?
Yes to both. Thank you.
Okay. Good afternoon. My name is Alan Andrew. I'm a master student at São Paulo State University. I would like to thank the organized committee of the SSR with the opportunity to present my data here. My study is entitled The Roles of Epididymal Protease Inhibitor in Terminal and C-terminal regions on the regulation of mouse sperm motility. So, what you need to know about EPPIN. EPPIN is a protein present in the testes and epididymis from mice and rodents. It's secreted by Searchable and epididymal epithelial cells and can be found on the sperm surface. The EPPIN plays 3 major roles. It modulates PSA activity, it possesses antibacterial activity, and regulates sperm motility. Though its crucial role on the sperm motility, it's considered a sperm target to development of new male contraceptives. It's known that EPPIN antibodies and small organic molecules that bind to the C-terminal regions, which contains the kinase domain, result in the inhibition of mouse sperm motility, both in vivo and in vitro conditions. But let us know about the effects of the N-terminal regions, which contains the WFDC region, on the sperm motility and EPPIN's physiological function. So our aim is to evaluate the effects of anti-EPPIN antibodies, mapping epitopes on EPPIN's N-terminals and C-terminals on mouse sperm motility. Here you can see a schematic representation of the primary sequence alignment between human and mouse EPPIN. Here is where the K20E antibody binds to EPPIN on the N-terminals region. And here is where the S21C antibody binds to the C-terminals region. To perform these experiments, we collect the mouse spermatozoa from cow epididymides and were incubated with pre-immune serum or S21C or K20E antibody and evaluated at 30, 60, and 120 minutes of incubation and evaluated by computer-assisted sperm analysis, where we obtain the sperm motility and the kinematics parameters VAP, VSL, VCL, AL8, straightness, linearity, the IRMI, and density that describe progressive and vigorous movements respectively. With this, we obtain the following results. Here, we observe that both antibodies, S21C and K20E antibodies, binds to the spermatozoa with similar conditions. We can see both antibodies on head and sperm flagellum. Then we evaluated the effects of the S21C antibodies, and we observed a decrease at 16 and 120 minutes of incubation of the motile spermatozoa when compared with pre-immune serum, and we observed a decrease at 30 and 60 minutes of incubation on progressive spermatozoa in an early manner. The effects of the S21C antibody was observed in the hyperactive spermatozoa in a late manner at 16 and 120 minutes of incubation. The same effect was observed in human spermatozoa. In the right side, we have the incubation with the K20E antibody at the same concentration, and we did not observe any significant difference. So we decided to test 2 different concentrations of 0.2 and 0.6 mg/ml, and we just observe an effect, a significant difference at concentration of 0.6 on the progressive spermatozoa at 30 and 60 minutes of incubation. Given that, we decided just to use the 0.6 mg/ml for your future analysis. Then we evaluated the kinematics parameters that describe progressive motility, and we observed that the S21C antibody affects the VIP with a decrease at 16 and 120 minutes of incubation, the VSL at 30 and 60 minutes, the straightness at 120 minutes with an increase. And when we incubate with a VAP, we observe just a significant difference at 30 minutes of incubation. To calculate the index of relative motility inhibition, IRMI, that describes progressive movement, we use the motility percentage and VSL, and we just observe the effects of the S2N1C antibody at 30 and 6 minutes with a decrease of the IRMI. Any significant difference is observed when we incubate with a K20E antibody. And then we evaluated the kinematics parameters that describe vigorous motility, and we observed in the upper graphics that the S2N1C antibody decreased the VSL at 60 minutes, the linearity with an increase on 120 minutes, and when we incubate with the K20E antibody, we observed a decrease on the AL8 at 30 and 6 minutes of incubation. To calculate the normalized density that describes vigorous movement, we use the VCL and the AL8, and we just observe a significant difference at 16 and 120 minutes of incubation. But when we look for the incubation with K20E antibody, we did not observe any significant difference. In conclusion, our results confirm the key roles of EPPIN C-terminal sequences on the regulation of sperm progressive and vigorous movements, and show evidence that EPPIN in terminal region can contribute to the regulation of sperm motility parameters. Further studies on EPPIN terminal and C-terminal domains may reveal novel insight into the underlying mechanisms involved in the inhibition of sperm motility, thus facilitating the development of novel EPPIN-bound male contraceptive compounds. I would like to thank the Laboratory for Reproductive Pharmacology and Andrology Group for my advisor, Eric Silva, and the collaborators and the financial support, FAPESP and CAPS, with the opportunity to develop these studies. If you have any questions, I'm open to listen, and I would like to thank for your time and attention.
Great, thank you. And what a wonderful animation there at the end.
Thank you.
We do have some questions for you. The first is, does the antibody affect the acrosome reaction, the ones that you're working with? Does it affect, or does it also have an effect on EPPIN's interaction with PSA?
Sorry, can you repeat the question?
Yes, does the antibody that you're working with affect the acrosome reaction, and does it affect the EPPIN's effect on PSA?
Yes, we are evaluating with your group these effects of if our antibodies can have an impact on the acrosome reaction. So we are testing some experiments to comprove this with our experimental model.
And for the interaction with PSA?
And with PSA, when I start to talk about the PSA in the start of my presentation, the EPPIN can modulate the PSA, so inhibition the acrosome reaction.
Great.
Because the inhibition of the acrosome reaction.
And there's a question about if EPPIN is present in other cells in addition to during spermatogenesis.
In spermatogenesis, how I said too before on my slides, he can be present on are secreted by certain cells, so it can be found there on the cell, inside the cells.
Okay. Are changes in sperm motility maintained mainly seem to be rather subtle? How much of a chance is there that the parameters would result in a fertility defect? Are there other parameters that would be more important than others?
Can you repeat more slowly, please?
Sure. So the changes in sperm motility seem to be rather subtle, small. How might the change result in a fertility defect, do you expect?
We observed these effects in our experimental model at the time of, how I said, at 120 minutes. And when we incubate with 240 minutes, we didn't know if the effect was of the EPPIN, because it's a long time of your incubation, and we observe just this effects just at the time of 120 minutes. I think that with this concentration is essential for affect the kinematics parameters and with this time.
Okay, thank you. I think we are running low on time, so I'll hand it over to Janice to introduce the next speaker.
Thanks so much, and thank you for that exciting update on EPPIN. Our next talk will take us to another part of the male reproductive track. So I'm pleased to introduce Noemia Mariani from Sao Paulo State University in Brazil as well, who will present work on recombinant seminal vesicle secreted protein SVS2 inhibiting the motility of mouse sperm.
Thank you, Janice. Can you see my presentation?
I can, looks great, thank you.
Okay, so good afternoon. My name is Noemia. I'm a PhD student at São Paulo State University, Brazil. And I would like to thank the organizing committee for the opportunity in presenting my data here. And the title of my presentation is Recombinant Seminal Vesicle Secreted Protein SVS2 Inhibits the Motility of Mouse Spermatozoa in Vitro. So the RAS proteins are secreted from the seminal vesicles and are the most abundant proteins in the semen of rodents and humans. These proteins play roles on formation of the semicoagulum, sperm function, and protection. The rest proteins in humans are seminogenin 1 and seminogenin 2. The seminogenin 1 binds to EPPIN on sperm surface, and this interaction leads to inhibition of sperm motility. In mice, the rest proteins are SVS. There are 8 SVS, and the SVS2 that is ortholog to human seminogenin 1 is involved in inhibition of sperm capacitation. And the effects of SVS2 are facilitated by SVS3. Recently, we found that SVS2 is an EPPIN binding protein on mouse spermatozoa. So these results support the hypothesis that seminogenin 1 and SVS2 are functionally conserved, but the effects of SVS2 on sperm motility are not known. Given that our hypothesis is that SVS2 acts as an inhibitory factor on mouse sperm motility, and that SVS3 facilitates the inhibitory effects of SVS2 on mouse sperm motility. For testing the hypothesis, we incubated spermatozoa from cold epididymis of adult mice in ATF medium containing increasing concentrations of recombinant murine SVS2 and or SVS3, using 1, 3, 5, and 10 micromolar of recombinant proteins. After 20 and 60 minutes, we evaluated the sperm motility by computer-assisted sperm analysis, classifying the spermatozoa in motile and static. In addition, we evaluated the kinematics parameters. So we evaluated the curvilinear velocity, straight line velocity, average velocity, amplitude of lateral head displacement, linearity, and straightness. First, we observed that SVS2 binds on the head and all the flagellum of mouse spermatozoa. Then we evaluated the effects of SVS2 on mouse sperm motility. So here we can see that SVS2 inhibits mouse sperm motility in a concentration-dependent manner, reaching significant at 10 micromolar. So 10 micromolar SVS2 reduced the motile, progressive, and hyperactivated spermatozoa. Consistently, 10 micromolar SVS2 reduced the average velocity, straight line velocity, and straightness in both times of incubation. 10 micromolar SVS2 also reduced the curvilinear velocity in both times of incubation, amplitude of lateral head in 20 minutes of incubation, and linearity in both times of incubation. So SVS2 reduced the parameters that describe the progressive and the vigorous movement. We performed the same experiments with SVS3. So here we can see that SVS3 binds to the head and only the meat piece of the flagellum of mouse spermatozoa. SVS3 did not affect the motile and the progressive spermatozoa. However, 3 micromolar SVS3 reduced the hyperactivated spermatozoa. Regarding the kinematics parameters, SVS3 only reduced the curvilinear velocity and the amplitude of lateral head displacement in 20 minutes of incubation, but SVS3 did not affect the other parameters. With your results, we decide to build a concentration response curve using IRMI parameter. So to calculate this parameter, we use the straight line velocity and the percentage of motility of spermatozoa with SVS2 or SVS3 in relation to the spermatozoa without the recombinant proteins. So here we can see a concentration response curve of SVS2 with IC50 of about 4 micromolar. Regarding SVS3, we could not build a concentration response curve because SVS3 did not change IRMI. For the co-incubation of SVS2 and SVS3, we choose 5 micromolar SVS2, the first concentration above the IC50 that did not change sperm motility. So we used SVS2 alone or in a molar ratio of 1:1 and 1:2 of SVS2 and SVS3 respectively. But the co-incubation of SVS2 and SVS3 did not show synergic effects on the sperm motility inhibition. So the motile, progressive, and hyperactivated spermatozoa were the same between the groups. In conclusion, our result provides additional evidence for the hypothesis that SVS2 and seminogenin 1 functions are conserved between rodents and the primates. Considering that seminogenin 1 and EPPIN interaction in humans leads to inhibition of sperm motility, our hypothesis is that SVS2 and EPPIN interaction in mice also leads to the same effect. And currently, we are testing this hypothesis. So we expect that these results will be important in the investigation of novel strategies for male contraception using mice as experimental models. This is our group and my advisor, Professor Eric Silva. To perform this study, we had financial support from FAPESP and the CAPES, and I would like to thank for your attention and time. So thank you very much.
Thank you very much. A few questions for you. The first is, does SVS2 affect sperm viability?
Viability, I don't know, but SVS2 has effects on capacitation. SVS2 inhibits the sperm capacitation, protects spermatozoa in uterus, but viability, I don't remember if has some study about this.
Okay. Do you think that SVS2 or SVS3 is a more promising contraceptive target?
I think that we are studying more about EPPIN, and since SVS2 binds to EPPIN, we think that exploring more the mechanisms of action that involve in EPPIN's functions, we can develop a new contraceptive. But I think that SVS2 is a promising, studying the mechanisms of these proteins is promising to male contraceptive development.
Great. And then the last question is concentrations, or the concentrations of the SVSU that you used, are they similar to the concentrations that you find in seminal fluids?
In seminal fluid, I remember that the endogenous concentration of SVS2 is 25 micromolar, but the stock concentration of our protein is 16 micromolar. So we use a concentration below of this, And we observed that 10 micromolar affect the mouse sperm motility, but is not similar to the endogenous concentration.
Okay, thank you. And I'll hand it back over to Janice for the last presentation.
Thank you so much. We will continue now with the last of the short talks selected from abstract submissions. And this one will take us into another very important area of contraception research, thinking about vaginal spermicides and microbicides. So it's a pleasure to welcome Nuch from University of Ottawa in Canada.
I'd like to thank the organizing committee for the opportunity to present our work, which is done in collaboration with Jonathan Angel, Gus Wong, Pramit Chet, and Rupert Call. The work presented is mainly done by Sung, Anna, Wongsagon, and Noparat. Our overall objective is to search for a single MPT reagent with spermicide and microbicide activity to be administered in the vagina. Historically, N9, a detergent spermicide, was investigated for microbicidal activity. However, its last clinical trial was halted halfway because women vaginally administered with N9 had higher HIV infections. So our approach is going the opposite way, searching for antimicrobial peptides with spermicidal activity. AMPs are cationic and amphipathic peptides less than 10 kDa, secreted as part of innate immunity. They exert microbicidal action by first binding to anionic lipids on the microbial cell membranes and then perforate it. We think the same mechanism would occur with sperm because they are enriched with an anionic sulfoglycolipid. We have focused on L37, the only human catalycidin with HCAP18 as its propeptide. It has microbicidal activity on over 50 bacteria and 10 viruses. This is a peptide sequence of L37 shown in comparison with GI20 and GF17, its truncated forms with similar microbicidal activity. The 3D structure of L37 is known. It is a helical, bent helical form with one phase enriched in cationic amino acid and the other hydrophobic residues. And these features allowed it to bind to the bacterial cell membrane and then perforate it. Another interesting thing about L37 is that seminal plasma has a high concentration of HCAP18, but when semen is deposited into the vagina, good motile sperm never see L37. This is because the processing of HCAP18 to L37 by gastrexin also in the seminal plasma occurs at pH 4. But seminal plasma has a high buffering capacity, neutralizing vaginal pH, and it takes 2 to 6 hours afterwards for the vagina to resume acidity, and that's when L37 is produced. And by that time, good motile sperm has swum into the uterine cavity already. So it looks like L37 is not good for motile sperm, especially when L37 could bind to SGG. Actually, when we treated partially or fully capacitated sperm with L37, GI20, or GF17, we could see the dose-dependent decreases of motility concurrently with the increases of membrane permeabilization. The spermicidal concentration of all three peptides on human sperm is 10.8 micromolar, and on mouse sperm is 3.6 micromolar. Note that these three peptides have the same helicity close to 100%. But when 3 amino acid of GF17 are substituted with the D amino acid, the percent helicity is decreased, and GF17D3 no longer has spermicidal activity. Like what happened in the bacterial cell membrane. Sperm, when treated with L37, shows severe damages on the surface membranes and even lost the acrosome. These three peptides inhibited in vivo fertilization. Females were injected transcervically with sperm plus and minus peptide. And 42 hours afterwards, they were killed and oviduct flushed to count 2 cell embryos plus unfertilized eggs. And this is to get percentage of in vivo fertilization. And in control females injected with sperm alone, this percent is more than 78. But in females injected with sperm and LD7 or GI20 or GF17, 0 fertilization. In this injection experiment, if the animals kept for 25 days, we could monitor the pregnancy. And we could see that females injected with sperm and LD7 could not get pregnant. But however, its reproductive tract tissues are histological normal. The LL37, GI20, and GF17 are prone to protease digestion. And this is why 17-by-Phe2 was engineered, having the same sequence as GF17, but 3 amino acids are replaced with the D amino acid, and 2 phenylalanines are replaced with biphenylalanines. 17-by-Phe2 is resistant to several proteases, and it has higher microbicidal activity on escape pathogens. We determined the spermicidal concentration of 17-by-Phe2 to be 21.6 micromolar for human sperm and 10.8 for mouse sperm. The higher concentration here than LL37 and GF17, because 17-by-Phe2 loses some alpha helicity. Regardless, female injected with sperm and 17-by-Phe2 could not become pregnant. And even though it's not a natural peptide, these mice did not show any abnormality on the vaginal histology. We compared degradation process of the 4 peptides in human cervical vaginal fluid, and we saw the lowest degradation rate with 17-by-Phe2. And when we resuspend sperm in CVF, we saw that 17-by-Phe2 at 32.4 micromolar, which is 1.5 times spermicidal concentration in the medium, completely inhibit motility. But for LL37 at 21.6, which is 2 times of spermicidal concentration in the medium, there's still 20% of motility there.
You've lost your audio, unfortunately.
Pardon?
I'm not sure if it's the Zoom or if it's the your computer.
What's happening?
Well, I could mostly hear her audio,
hear
so
her
I don't
fine.
know if it's machine by machine. Anyway, Nooch, please continue and wrap up.
Okay, we looked at microbicidal activities of the 4 peptides, and we have shown that LL37, GF17, and 17-by-Phe2 have a preferential microbicidal activity on the STI-induced Neisseria gonorrhea over the commensal bacteria Lactobacillus crispus. On HIV study, we first showed that 17-by-Phe2 is an effective peptide to inhibit HIV replication and infection in ACH2 cells. We therefore treated HIV with 17-by-Phe2 and used it for inoculate PBMC. And we could see the inhibition on HIV infection here. This experiment is still ongoing. However, with all of these results, we think that both L37 and 17-by-Phe2 are promising candidates to be developed into NPT reagents. Thank you for your attention.
Thank you very much. And what an exciting way to end talking about multi-purpose preventive technologies. A couple of questions for you. Did you look at other endpoints in the vaginal health other than histology to ensure there was no impact?
Yes, we have experiments ongoing. I have another slide there actually. We are injecting mice 3 times, 3 consecutive reproductive cycle with LL37, and then we're looking at vaginal microbiota. We get the result from 1 experiment already, and there's no changes. But we have VCF, vaginal contraceptive form, which is 12.5% N9 as a positive control. And we saw some change there. We look at the cytokines, chemokines too. There's 7 of them that we looked at and no changes, but VCF increased MCP1 expression.
That's very promising. The next question is, human contraceptive gels can enter the uterus during intercourse. Did you examine the mouse uterine endometrial histology to determine if there was any disruption due to the compounds?
Say that again.
The contraceptive gels can get into the uterus during intercourse. So did you examine the mouse uterine endometrial histology to see if there was any disruption?
Yes, yes. We published the results already in Human Reproduction. In the first paper is in 2014, and the second paper in 2018. No changes. No.
Very good. I think that is all the questions that we have. And I think we're a little over on time anyway. So I'll hand back to Janice to close out.
Thank you.
Yeah, and thank you very much for those presentations. Thank you all for attending. I'll remind everyone that we do have a virtual happy hour on October 15th, which is the conclusion of this three-session webinar on contraceptive development, and that's when we'll be announcing the winners of the oral and poster presentations. Also, a heads up, you'll be receiving an email shortly after this webinar concludes to provide feedback for us at SSR. So we very much appreciate you completing the poll so that we can serve our members and other conference attendees the best we possibly can. So I want to thank again the organizing committee who worked very hard, especially Daryl Russell, our fearless leader as chair, all our wonderful speakers and our guest judges. And I look forward to seeing everyone a week from today for our next session. Take care, everyone, and thanks so much for joining us.
Thank you.
Bye-bye, Janice. Thank you.
Thank you. Bye.